It's a pleasure to us to present partial result of our research carried out in Chile during pre-season. The title of the talk is Bioactive Compound in Fruits of Wild Maqui, Aristotelia chilensis, in different ripening state and environment. This research is found by Native Forest Program of CONAF. The authors are Antonio Pinto, Verónica Fuentes de Alba, María Dolores López, and the speaker Susana Fischer. This presentation is organized as follows: introduction, objective, methodology, result and discussion, and conclusion finally. Maqui is a small tree here, and it's an endemic species of the South Antarctica forest of Chile and Argentina. It produces small berry with high antioxidant content. It is distributed in semi-arid climate zone in the north part of Chile, in the region of Coquimbo, and in the south part of Chile, in Aysén region, in a temperate climate zone. It's a species that we can approximately find 170,000 hectares of maqui, mostly with wild stand. Maqui is considered a pioneer species because usually it's the first species to colonize burned forest or abandoned soil, and it can develop in place with edaphic problem associated to erosion. In this sense, it is considered an important agent for soil conservation with a great capacity of regeneration and to generate conditions that favor the establishment of other species. It can tolerate short drought period of less than 1 month and can grow in any exposure and various slope range. For the Mapuche native culture, maqui is one of the 3 sacred plant species, a symbol of good intention, and is used in popular medicine. They are careful with the harvest to protect the tree. Also, maqui harvest is an important summer source of income for many people in rural areas who collect non-wood forest product in the wild. And for naturally, the current harvest practice of maqui is by cutting the whole fruit behind branch, resulting in serious environmental damage and unsustainable practice. The maqui fruits are considered as one of the berry species in nature with highest level of polyphenol, tannin, and anthocyanin in comparison with other berries. Maqui has been referred as a superfruit due to their phytochemical composition and antioxidant activity. The most important of maqui berry lies in its benefits to the human health, not only due to its extraordinary antioxidant capacity related to phenolic compound, but also due its anti-inflammatory, antimicrobial, anti-cancer, and nutritional property, among others. Therefore, it's a great raw material for supplement and pharmaceutical products. Given its beneficial property, the maqui is a berry with high demanded by the international market. However, as production and export come mainly from wild collected fruit, it is supplied a very little amount. Currently, most volume are behind exported as powder, dry, frozen, juice, and preserved maqui fruits. Since maqui production come mainly from harvesting fruit from wild stand, its availability is less than the growing demand, and the bioactive compound content are influenced by collection time, ecotype, and environment. There is very little information on the management of wild maki for sustainable production. Therefore, knowledge of the production management is crucial to develop sustainable management program. Given the present background, the objective of our research was evaluate the variation of the polyphenolic compound, the antioxidant capacity, and the fruit quality in 3 states of maturity in 3 locations in the General Valley in Chile, exposed to a Mediterranean and temperate climate. We harvest the maki fruits in 3 locations: Tehualemu, Cayumanqui, Coihueco. Where are these locations? All are in the Central Valley. Tehualemu in the coastal plain, Cayumanqui in longitudinal valley, and Coihueco in the fall of the Andean mountain. Fruit was harvested in summertime between December to January during 3 consecutive seasons in 6 selected stands. 3 states of maturity were harvested: ripe, coloring, and green stage. Samples of maki were ground in a porcelain mortar and then treated with 5 ml of methanol, water, and acid formic. Each sample was kept in ultrasonic 2 times, centrifugate, and finally, the supernatant was kept in amber flash for later analysis. The content of polyphenol was determined by the reduction of Foligno-Culture reagent. The total anthocyanin was determined using the different pH tests, and the free radical scavenging activity of the sample were determined using the DPPH method with modification. For our result in the estimation of total polyphenol, in this table, you can see that the highest total polyphenol content are observed in the season 2020-2021, while the value fluctuating from 2,218 to 1,040 milligram acid gallic equivalent in the ripe state. In the coloring state, the values are between 1,808 to 1,730. And in the green state, the value are 2,068 to 1,730 milligram acid gallic equivalent. The highest value were recorded at the ripe fruit state in Coihueco and Cayumanqui in both seasons, while Tehualemu did not share the same values. The lowest polyphenol content were observed in the green fruit state. For determination of the total anthocyanic content, we can see that the higher content was during the 1920 season, and where the high value was 1,130 milligram of cyanidin triglucose in 100 milligram fruit, while the lowest value was 1.3. This variation is directly related to fruit development state. Thus, the higher concentration are observed at the ripe fruit state, while the lowest value are observed at the green fruit state in all the study where we made. In general, Coihueco and Cayumanqui recorded the highest concentration, and Tehualemu the lowest. For the antioxidant capacity in the fruit harvested, the value varied between both seasons. It was higher in the second season, 2021. Cayumanqui recorded the highest value in both seasons, but not at the same fruit development stage. Tehualemu record higher value at the fruit coloring state compared to those observed at the red fruit state in both seasons. In the 2021 season, the antioxidant capacity reached a similar value around 70,924 at coloring fruit and grapefruit state respectively. This situation was similar in Coihueco. In Tehualemu, however, the highest antioxidant capacity was observed in green fruit state. Conclusion. The results are agreed with those reported in other studies and other berries in which the total anthocyanin content increased as fruit development progressed from the green fruit state to ripe fruit state. There was a direct relationship between color change and fruits and the content of anthocyanin and total polyphenols. The level of total polyphenols observed in the maqui cultivars under study in the pre-state site and are higher than those observed in other berries such as blueberry. Variation of total polyphenol and anthocyanin between season and study site can be explained by differences in the edapoclimatic condition. The antioxidant capacity was lower in Tehualemu compared to Cayumanqui and Coihueco. Many thanks